In the name of God, hope you all to be healthy and doing well. We are in endocrine physiology subject, and in this file, I'm going to start to discuss about the pancreas and the hormones that are released from the pancreas. You know that pancreas is not just releasing one hormone, but the most important hormone that is released in the body is insulin, and insulin is a product of the cells in the pancreas. When we want to talk about the pancreas, we should give you an overview, anatomical overview of the pancreas. And as you see in this figure, pancreas is a part of the gastrointestinal system, or is a partner for the gastrointestinal system. It has two parts. One part is named as exocrine portion or part that release the enzymes that are needed for the normal digestion of the nutrient in the gastrointestinal system. And as I explained you about these enzymes and the function of this enzyme in the gastrointestinal physiology, without this enzyme, gastrointestinal system is not able to digest the proteins, carbohydrates, fatty acid, nucleic acid, and in this way, pancreas is an essential part of the life. And without this part and without the function of pancreas, life is impossible. On the other hand, between these exocrine parts, we have some other cells that do not release any enzymes and do not have any work with the gastrointestinal system. These population of cells that are distributed around the pancreas, particularly between the exocrine part, they are dispersed between the exocrine part. These population of cells are named as the pancreatic islets, and because of their discovery and the scientists that discovered this islet, these islets are named as Langerhans islets. If you have a look at these islets in a schematic view, you can see that between the several exocrine cells and ducts that you can see in these figures, we have the Langerhans islets, and in the Langerhans islet, there is not just one type of cell. There are several type of cells that are gathered together, and in this figure they are shown in different colors. As you can see, the yellow cells are named as alpha cells. Alpha cells release a hormone named as glucagon, and glucagon is an opposite hormone for the insulin, and I will discuss about this. Another type of cell is named as D cells. These cells that the population is not very high, but these cells that are shown in this figure here, and these cells are releasing a hormone named as somatostatin. And finally, the most abundant cell type that we can see in the Langerhans islet are named as beta cells. Beta cells are responsible for releasing the most important hormone and the most famous hormone in the body, insulin. They also release another type of hormone named as amylin. Among these cells and among the hormones that are released from these pancreatic islets, as I told you, insulin has the most famous functions, and several years of discovery lead to the discovery of this hormone. but the other hormones are also participating in the function of the metabolic control in the body in cooperation with the insulin. This figure shows the different, in the right side, you can see the population of the cell that have different type of cells and different staining characteristic that you can see in this figure. Okay, let's go forward. This figure show the different type of cells that I told you in immunochemical studies, and you can see the colors that red colors are insulin and green ones glucagon. And these are the most frequent hormones that are released from the pancreatic isolates. If I want to have a brief historical view about the Langerhans isolate, and their discovery returned to the 19th century when Langerhans, for the first time, described these isolate in 1869. And as I told you, for this reason, these isolates are named as the Langerhans isolates. At the same century, and even before, it was known that that the removal of the pancreas from the body will cause the picture of diabetic patients, because the diabetes was known for a long time, and therefore it was discovered that the pancreatic, when the dogs are pancreatectomized and the pancreas is removed from the body, they will develop fatal diabetes, and therefore they conclude that one thing is inside the pancreas that is responsible for that its that its shortage and or its hyposecretion is responsible for diabetes. And thereafter, there was several studies that were trying to extract to homogenize a normal pancreas and extract the juice, and then inject the juice to a diabetic animal. They were thinking that in the juice, in the extract, there is the factor that is responsible for diabetes. And all the time, these studies were failing because this factor was not working. In about 100 years ago, two scientists named as Banting and Best for the first time realized that when we homogenize all pancreas together, pancreas also contains some proteolytic enzymes, and therefore they conclude that maybe that factor that we are seeking may be destroyed by the exocrine hormones. Therefore, they develop one animal that firstly, the exocrine part of the pancreas is destroyed by blocking the OD sphincter and creating a situation of acute pancreatitis. Therefore, the exocrine part was destroyed, and then this pancreas was homogenized, and then the extract was injected into a diabetic animal, and then they discovered that this factor is working. And thereafter, this factor was named as islatin and then insulin. This was the discovery, the moment of discovery of insulin, and you know that 1 year later, it was awarded awarded Nobel Prize for this discovery. Thereafter, there was several studies to determine the primary structure of the insulin, and in 1954, Sanger was successful in the determination of the structure, and again this discovery was awarded by Nobel Prize. This shows how much this insulin was important for us. The pancreas developed during the embryonic life at the week at the fifth week, and then will develop into a normal one during the rest of the embryonic life. It is derived from pancreatic duct and endodermal origin. As I told you, the islet of Langerhans are distributed around in the pancreas, mainly in the body, tail, and anterior portion of the head. The population of cell, 60% is beta cell, 10% alpha cell, and the other cells such as F cell, D cells form the remaining. And the product, as I told you, insulin, amylin, GABA, glucagon, somatostatin, phospholipid P are also the other products that are released. The islets receive a very high circulation because they release several hormones. Therefore, they need high blood circulation, not because of the oxygen delivery or nutrient delivery. They need this type of blood flow because they want to send the hormones into the blood. And this figure show a type of the blood flow that we have in the islets, and some graphs, some histological graph that show the islets. Okay, let's start with this introduction. It was a little lengthy, but with this introduction, let me to start discussing about insulin, the main hormone of the islet of pancreas. It is accepted that insulin is the dominant metabolic regulator hormone. I do not mean that the only hormone that is responsible for regulation of the metabolism is insulin, but insulin is the most important one and dominant factor. The synthesis and processing of insulin inside the beta cell is happening. Firstly, a very large gene that contains the length of insulin, also the coding of insulin will be expressed firstly, then alternative splicing will happen, and finally a large protein named as proinsulin will form. Proinsulin is composed of three segments, A, B, and C. Finally, inside the vesicles and granules, and under the function of the enzymes, B and A segment will be separated from the C segment. And then these two A and B segment will be connected to each other, and therefore the final hormone will form. And the C peptide, the C connecting peptide, will be released in as a co-peptide, as a side product of the production of the insulin. This figure shows the B chain, C peptide, and A chain, and then the C peptide is being removed. A and the B chain will connect to each other by some disulfide bridges, and then C peptide also will be released separately. When you have a look at the difference between the A chain and B chain in different animals and different species, you can see that there are some differences between different animals. For example, as you can see in the pig and dog, at the at the A chain position, at position number 8, 9, and 10, there is some difference. For example, in pig, dog, and rabbit, these are the same. But in cattle, alanine is substituted, is replaced with threonine, and also in horse. In position number 9, you can see that in goat, glycine is replaced with serine. And in position number 10, again, you can see that valine is replaced in cattle and goat instead of the amino acid that we have in the pig and ducks. Have a look at the B chain. Again, you can see that at the position number 30, there is some difference. In pig and dog, it is alanine, but in the rabbit, it is serine. And in horse, it is serine. But in the cattle, goat, sheep, pig, and dog, it is alanine. This shows that we have some different isoforms of insulin according to the species. And for this reason, sometimes injecting the insulin derived from one animal to another animal can cause immunological responses. This figure shows the number of the amino acids. As you can see in the chain B, we have 30 amino acids, and in the chain A, we have 21. And the C-peptide that is removed from the final insulin. When insulin enters the blood, it is able to dissolve in the blood and distribute all around the body. The insulin is a hormone that is known as a multiple target hormone. And for this reason, we can say that most cells in the body express the receptor for insulin. And receptor for insulin is a tyrosine kinase receptor, belong to the tyrosine kinase receptor family, that it has two alpha subunit and two beta subunit, that when insulin connected bond to them, they form that they are dimerized and then activate I'm not going to discuss about the biochemical events inside the cell, but briefly start to activate a very important downstream molecule such as IRS, that is named as insulin receptor substrate. It will be phosphorylated. Then this phosphorylated IRS will stimulate the other molecule inside the cell, and finally produce the functions and effect of insulin. Those effects that I will discuss about. It affects the metabolism of glucose, metabolism of fat, metabolism of protein. It affects growth and affects the gene expression of several other proteins. And all these effects are produced by these by these receptors and these enzymes. For this reason, if for any reason these receptors were not working, if for any reason these receptors were failed to produce this effect, even though that insulin is available, but we say that we have the case of, or the state of, insulin resistance. And insulin resistance means that insulin is available, but cells are not able to respond to that. This figure shows the right figure show the connectivity and dependency that we have between insulin and glucose concentration. As you can see, glucose concentration normally is in the blood should remain around 100, fast blood glucose, around 100 milligram per deciliter. When these glucose concentration increase from 100 to, for example, 150, as you can see, the insulin secretion is increasing several times. And when it reached 200, for instance here, when it reached 200, as you can see, it reached about 8 times. When the blood glucose reached about 300, the insulin secretion even increased up to 15 times. What is the meaning of this figure? This figure shows that insulin has a very, very high sensitivity to the glucose concentration. Every time that glucose concentration want to increase, immediately insulin will be released and try to return it back to its normal level. On the other hand, every time that glucose concentration is decreased, then insulin secretion will decrease. And you should keep in mind that insulin secretion was not zero at all time. We have a continuous secretion of insulin every time that glucose concentration is increasing. Therefore, insulin also will increase. And every time that glucose concentration is decreasing, insulin will decrease. And by this way, insulin is able to control the glucose concentration in 2 ways. One, both increasing glucose and decreasing glucose. Insulin is able to decrease the glucose concentration by increasing its concentration. But if we inhibit the baseline secretion of insulin, then we are able to increase glucose concentration by decreasing glucose uptake by the cells. The response of insulin to glucose change it has two phases. There is a rapid phase that is happening in some minute, and it is very, very strong and immediate. But over some minute, it returns back to not to a normal level, but lower level. And over time, it again shows a late response. Therefore, we have a fast response and a late response. This shows if glucose concentration in the blood is increasing, there are lots of insulin that are available and synthesized before inside the beta cells, and these stores of insulin will be released into the blood. But next, over time, this storage will be depleted. Therefore, these will return to lower concentration, and over time, new insulin will be synthesized and released. Therefore, over 60 minutes to 2 hours, or I can say in 1 hour to 2 hours, the concentration of insulin again return to very high levels. This is the new insulins that are produced now, and the acute and very fast phase was those insulin that were stored before. This is the mode of response of insulin. And if we ingest a high glucose food and suddenly increase the blood glucose, sometimes the workload for the pancreas will increase, and it may cause some problem for it. For this reason, we say that drinking sodas that have very simple sugars and they are easily absorbed into the blood may eventually cause exhaustion of the beta cells. Okay, this is showing the insulin secretion that is biphasic. This figure is the same, but with more detail, you can see the concentration of glucose and the insulin secretion, concentration of the insulin secretion. As you can see, increasing glucose concentration immediately increase. And you can see that in some area, it has a very high sensitivity that we can see in the steepness of the line. And the other figure is showing the biphasic release of insulin that I explained about it. This shows the receptor of insulin that I told you. It has two subunit, two alpha subunit, two beta subunit, and they are dimerized and then produce the response that I told you about it. And this shows the molecular events that I explained about it. They will be activated, and these signaling pathways eventually cause the responses that I will tell you about them, particularly increasing glucose uptake that I will explain about it in a few minutes. The rate of response to insulin also is different. That slide that I told you was the release of insulin, that was biphasic. The response of insulin also has a rapid phase. Rapid phase means that insulin do not need any new protein to produce. Stimulate the insertion of the pre-produced proteins and channels and carriers to the membrane. Therefore, increase the membrane permeability to glucose, for example, immediately. This is very rapid and do not need very much time to produce. Some effect of insulin are intermediate. They are not very rapid, not very slow. These functions are mostly metabolic functions. For example, stimulating the function of one enzyme, stimulation converting glucose into glucose 6-phosphate and phosphorylation into energy mediators, energy intermediates. These metabolic functions that I will explain about them are intermediate, and one part of the insulin response are delayed. These in effect need time. For example, stimulating the expression of several genes that should be expressed and then translated into new proteins, and new proteins help the change in the structure of cells, growth of the cells. These growth and gene expression effect that we have from insulin, and they are very important, they are not very rapid. They need several hours to produce, and they are delayed. Therefore, if I ask you about the response mode of insulin, we have three modes of response: rapid, intermediate, and delayed. Now, let me to discuss about and explain you about the mechanism of insulin secretion. Because I want the file's length to be manageable, therefore I explain the mechanism of secretion of insulin in the file number 11. This was the file number 10 that here I finished it, and the mechanism of secretion will be explained in the next file.